Great. Okay, same as always, we're gonna start this side, go around the table, and hit a loop-de-loop. Any questions you have from lecture this week, from the discussion worksheet that maybe didn't get addressed in discussion? We're trying to get ready for exam 2, so let's get it started.
I'm just here to listen.
Okay, listening is good. That's also important. Next person.
I was wondering if you could go over the experimental step up for question number 5 from last week's discussion. It is not.
It is not. You just gotta tell me what question number 5 is. I don't have that type of memory.
No, it's
Maybe just like, yeah.
Which discussion was this from?
Last week.
Oh.
Lots of discussion. Okay, telomerase question set up. Okay. So first things first, we are going to get a cell lysate. It basically has intact protein that is active. And the whole point of this experiment is to see whether or not there's active telomerase as part of this group of protein that we're getting from the cells. So that's the first thing that's in here. The next thing that we have is DNA. And sometimes you'll hear it called a DNA primer. What they mean by that is that you have DNA that has that 3' overhang, meaning that it is primed and ready to be extended by telomerase. Okay, so that's clear. So basically what they end up doing is mixing this together in vitro. And then if there's telomerase activity, you're going to extend these based off of all the repeats from the telomerase. So if there's telomerase, you're gonna extend it. The thing about telomerase is that for every species, there is an RNA primer already within the telomerase, RNA primer. This primer is a set sequence and it's a set number of nucleotides. So there's so many repeats they can add on. So when you take something where you see telomerase activity and you run it on a gel, depending on which DNA it started adding to, you have different numbers of repeats. So when you run it out on a gel, you're going to have different lengths, shorter, longer, of DNA. And what the gel is showing is just all the DNA product. Does that make sense? Okay, so now that we know how the experiment works, let's talk about different lanes, things that we put in there. So this is going to come back to what we talked about in class today with positive and negative controls. Telomerase. What types of cells express telomerase? Stem cells. Yep, stem cells, cancer cells, germ cells. All these will have telomerase, which means on a gel, we should be able to see telomerase activity. So for example, on the worksheet, you have 3 lanes for each of these. I'm just going to start with number 10, 11, 12. 13, 14, 15. I can't, right? 13, 14, 15. And then, so for stem cells and cancer cells, we expect there to be telomerase activity, meaning we should see these repeating units or evidence of repeating units from the telomerase activity. So we see that in 2 of the lanes. If you read the figure legend, you see that the figure legend tells us that lanes 10, 11, or 13, 14, whatever are the duplicates, or whatever are next to each other, or the first 2 lanes are duplicates. So you're just running the experiment technically to make sure I get the same thing each time. The 3rd lane, they said, is an additional control, where before they did this whole entire thing, they heated it up, right? Is that what it says? What would happen if you heated up a protein lysate? Yes, denatures, and it denatures the telomerase. If we denature telomerase, do we have activity? If we don't have activity, do we get extension of DNA? If there's no extension of DNA, do we expect to see stuff on the gel? So no stuff on the gel. So that's what those controls say. Now, another part of the question, it is asking or saying the goal is to take some skin cells, turn them into stem cells, right? So this very first lane, we have skin cells. And I think this is numbers 1, 2, and 3. We don't see anything. Why? Skin cells don't have telomerase activity. So you don't see anything regardless, whether it's the experimental, or everything's intact and set up properly, or whether you denature the protein or telomerase before time. So what does this serve as in our experiment? What type of control is this? Negative control, exactly. Then in the middle, you have two unknowns. This is 4, 5, and 6. 7, 8, 9. No, I can't count. Okay. And then we see activity in 7 and 8. So these are the samples where we try to take the skin cells, make them into stem cells. So we're trying to make them into stem cells, and we know that stem cells plus have telomerase activity. What types of controls are stem cells and cancer cells? Positive control. So now that we have a clear negative control, clear positive controls, the additional technical controls would be like, oh yeah, this is specific to activity from telomerase. What can we conclude about samples 1 and 2? So if they have telomerase activity, we assume that we're able to take skin cells and make them more stem-like. Okay, so now is that clear? You're making the face. You should,
I thought, it was a positive negative control, it's why the telomerase would result in the DNA being broken into different size fragments that get repeated.
so not broken. So when we start out, well, yeah, it's gonna be created, and that will clarify things. When we start out, this lysate has no DNA in it. We're isolating only the proteins. Then the only DNA we're gonna have is the DNA that we add. Make sense? So we're starting off with a small amount of DNA. If we ran small DNA by itself, it would only be at the bottom. But then when we get telomerase activity, we're making the DNA longer and longer and longer. So it's a period.
All the way.
Mhm. Okay, cool.
That makes sense.
Yeah, if you ever feel confused, like don't let yourself be confused. There's no reason, especially I'm standing right here.
That makes sense now, thank you.
Like radius has caused like telomerase, why it's obviously been going up and up.
Yes. Okay, any other questions on this particular problem? Yes,
So I understand the, like, why of the third main thing, I think we did it, is because of the policy and specificity. I'm kind of confused, like, what is there to.
so this is actually not a comparison. It's just,
Or, like,
it's
what was there for this?
why it's there, right?
Yeah.
It's so you have your regular negative control or your positive control. And a lot of times in an assay, we add in what we call technical controls, just to ensure that the assay is measuring what we think it's measuring.
Oh, I see, I see.
Yeah,
So
so that
it's-
enzymatic activity was necessary.
Okay.
Exactly, yes.
I have a question. So the negative, the skin cells is a negative control.
Mhm.
Samples 1 and 2 are experimental conditions. Cancer cells and stem cells are positive control. And the third lane that we see is an additional negative control. That see like when we remove that protein, we don't have it. It's dependent on telomerase or whatnot that we're not getting activity.
Exactly.
So it's also a negative.
Would it be negative or would it be positive because it's like a treatment to that thing?
So usually what I call this, when you see something like this, I call it an additional technical control. It's making sure the actual like technique is working and due to what we think it is.
So I should call it that.
Yeah.
So.
So yeah, it's not technically wrong, and then like on an exam, it'd be really clear what you're asking, what I'm asking. Yeah, so it's all like, oh, awesome semantics. But negative control, positive control, and additional control to ensure that this is caused by protein enzymatic activity or protein activity. Okay, so for example, so say I wanted to make up another control. What if I decided to use RNase in this experiment? Like, what would that do?
Was it all years also?
Mm-hmm.
You would see no.
No extension. And so think about how this whole experiment works. What is in telomerase? So RNA primer, if you added RNase, you lose the primer, you don't get extension. It's another way to add a control. Okay, so we're clear on this experiment? Okay, cool. Now over here.
If you point out a new temperature related like to melting temperature.
They're essentially the same thing. The word annealing is used when we're talking about in vitro PCR.
Or you could have a lower melting temperature or it only a little like a lower temperature.
Exactly.
No, I'll put it on the primary combined, but so that the strands don't come down. Together.
So in PCR, we set the melting temperature by how we designed the primer. And the melting temperature is the temperature of the actual strand and how it interacts with the DNA. The annealing temperature is the temperature at which we run the cycle where we expect the primer to interact with the DNA. So you want the annealing temperature and the melting temperature essentially to be the same. That's what our goal is. So think of like melting temperature characteristic of the primer itself. Annealing temperature is the temperature that you're choosing to run the thermocycler at for the annealing to happen. Yes.
Yeah, that's confusing, because that's like the melting temperature, that's the temperature, if you raise it to separate the strand, right?
So you raise it past the melting temperature.
Yeah, right.
So the melting temperature is where 50% gets put in half.
Okay, so yes, and then, yeah, you want to raise it, and then when you lower it to the annealing temperature,
Yeah, so we lower it to the annealing temperature, which is usually a little bit below the melting T.
yeah. Okay, that's why I was like, using the fire. Same.
Yeah, it's usually about the same. So say, we tend to like run PCR cycles at like 60 degrees for the annealing temperature. Design primers that have a annealing temperature of about 60 degrees
Okay, so you're just associating the melting temperature with primary.
with the primer.
The annealing temperature, the actual temperature of the primary is doing the activity.
Yeah.
Like being standard.
Yeah, so I'd almost think of the annealing temperature of what you're setting the instrument to. The melting temperature is characteristic of the actual primer. And we're trying to get them in a range so that we can just run the experiment through the cycles. Here, any question?
Oh, yeah, I know it, one kind of going back to the lecture from last week, it was regarding the PCR annealing temperature. Just want to clarify that I heard what you said, right? So this is, why the correct annealing temperature means specific primer binding, but lower annealing temperatures result in non-specific primer binding. So what I wrote is just right. So primers with specific GC sequence that requires specific temperature to bind. We want primers to anneal to DNA before DNA anneals back to the other DNA strand. Therefore, we need primer with high GC content, but a high annealing temperature. So if temperature is too low, we will have less specificity, primer binding with primer, DNA binding with DNA, which is no good.
Yes, exactly.
I have other questions. too.
I mean, you can go for a real question. That was just like,
Oh, yeah,
you just said you knew things and then you did. So-
yeah, so it was kind of going through the three different mutation examples, which was like. Kind of going like, okay, like cytosine going to uracil is
Mhm,
like very common.
yeah,
The uracil then eventually gets repaired to thymine, and this is what we call a transition mutation, correct?
yes,
And then the next part, which was the 5-methyl cytosine to a thymine. Once it goes to thymine, then is that in essence, does anything happen to that? When the 5-methyl cytosine becomes thymine, does anything else happen to that thymine, or that's it, that's the transition mutation?
so that's it,
Oh, that's it.
because does DNA recognize thymine as something bad?
No.
No, it's a regular base, and then so on the other side, when it replicates, it'll just stick an A over there.
Then
Yeah,
last,
done.
oh,
Okay, so transition mutation, two ways to get to it. You have deamination of, oh my god, what letter is this?
here's.
No.
5-methyl cytosine.
There we go. Or CDU. So either way, both of these require replication in order for the mutation to actually like set in and be heritable. But you have to have replication. But the deamination of 5-methyl cytosine to a thymine results in a transition mutation. If you have deamination of cytosine to uracil, then you have rounds of replication. You end up pairing with an A, and then after you do base excision repair, you change this to a T, and then you still end up with a transition mutation.
And then the last example was the one where you have these, the oxidation of nucleoside bases where, going from guanine to aoxoguanine, and aoxoguanine disrupts DNA shape. And I believe, if I remember correctly, it combines with cytosine, which is normal, or it combines with adenine, which is not normal. And when it binds to adenine, that's a transversion mutation.
Yes.
Okay.
So you got a 50-50 chance of this occurring, and then if it does this, it's the transversion.
And I have other questions.
Okay, so come around and we'll come back.
So for PCR and like making the primers, can you explain like the differences between like on the left side of the strand and the?
Yeah. Okay, we're just gonna make up a strand and we are going to design primers because like I swear, if we don't get 100% correct answers on this on the exam, my heart will break into a million tiny pieces. Okay, so we're gonna go 5 prime. We'll say A, T, C, G, 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 C, A, T, G, and 3 prime. So the whole idea when we want to design a primer amplified DNA is that we are choosing a target that we want to amplify. So, for example, say I want to amplify what's from here to here and nothing else. This is my target. So the goal of designing a primer is to make sure that I can amplify only this target DNA. Meaning that when I design the primer, I don't want it to synthesize anything on the outside. So when we think of DNA, remember there's always a complementary strand. But when designing a primer, I usually say just write out the complementary strand. So we're going to go 3 prime. This is going to be T, A, G, C, 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 G, T, T, A, C, 5 prime. And so in order to make sure that I'm amplifying only this piece, I need a primer that will go from 5 prime to 3 prime this way. Also a primer that will go from 5 prime to 3 prime this way. And usually if you write it in between, it makes a lot more sense. So I want to design a primer that is going to be here, and then a primer that is going to be here. 5 prime. Yeah. 5 prime. There we go. So you can see how on the left side of this will be my primer 1. But the primer 1 is actually complementary to the opposite strand. And then the primer 2 is going to be complement to what I have written here. And remember that anytime you write DNA, you have to write from 5 prime to 3 prime, 5 prime to 3 prime. The reason being, this is how DNA is synthesized. If you go in order of primer, they need it in 5 prime to 3 prime. It's the only way it's read, it's the only way it's made. So it doesn't matter what strand it's on, you have to write it 5 prime to 3 prime. So for this primer, because I want to extend this way to replicate the strand, I'm going to have A, T, C, G, G. And essentially it looks just like my original strand. So the shortcut for primer 1 is just to write exactly what you have here at the beginning to the left. That's a shortcut for primer 1. And then primer 2, remember we are writing this so we can extend from 5 prime to 3 prime. We're just going to write the reverse complement. So the reverse complement is already written here for us. And then so instead of writing it this way, we're just going to write it the other way. So primer 2 will be 5 prime C, A, T, T, G. So think primer 1 is the same as the strand that we have in front of us. Primer 2 is the reverse complement of what we have written down.
Basically, you always know the trend, like you can pretty much always get it.
You can always get it. Like I said, get this right. Get this right on the exam, please. Okay, any questions about primer design? Okay, here. Wait, are we? There's. Oh, okay. I skipped like 5.
Like,
Burner. Okay, so sorry.
so, okay, so. Burn is like the one who's that base, right?
Yeah.
The name says base. It's for nucleotide. But it's only for one nucleotide, and then. Nern is for like multiple nucleotides, and then there's one nucleotide in the name, so that is not really.
Keep it straight in your head,
Yeah.
right? So what I suggest is writing yourself a little cheat sheet. Organize your notes in a way that makes sense so you know which ones there are for and in what scenarios. So we have mismatch repair. We'll start here. Mismatch repair. This is 1 nucleotide. Basic excision repair, 1 nucleotide. Nucleotide excision repair, multi. You can already see how like I'm making a little cheat sheet for myself. So what type of damage do I have to get for mismatch repair? Where does this mutation or mess up come from? Replication. So DNA pole effed up. So replication error. Where does this one come from? What mutations do we need to do basic excision repair? Come on, y'all, you got this.
Well, they're like this. One. It's like, it does, do an excision, but not really, like, there are the ones that does the enzyme.
Mhm.
And then, that one I don't remember. What it is.
Okay, so let's go for basic excision. Something's wrong with the actual base.
Oh, they like this.
Yep, the glycosidic leakage gets cut in this repair. So the types of damages that occur are something that impacts only the base. So this would be like deamination, or oxidation, things like that. And then for nucleotide excision repair, what type of damage do we have? Or what causes it? Or what do you want to repair with nucleotide excision repair? Yeah, adducts or dimers, so UV induced, like TT dimers, where the polymerase gets stalled and it's big. They have to cut out a big piece. Like, know the differences between, know what type of mutation it fixes, where those type of mutations come from. Know how the glycosylases work, what's the glycosylase doing? Cuts out the base, then makes like an A basic site. Okay, are we clear for these three? No problem. Now we're here. Oh,
You see now, I had it all set up and then.
it'll come back, it'll come back.
Sorry.
They don't gotta apologize. I was gonna say it's Friday. Oh my God, it's not Friday. Oh my gosh, almost Friday. Okay. Ooh, discussion question.
It's basically just like. For the patient, to like, for instance, this is testing the monitor because in our like. And, I guess I was kind of confused on like into these things. And like specifically for this discussion paper because I understood the one in lecture, but I was gonna confuse it.
You understand all of them. I want to get up on you. To give you what you do. Okay, how many of you have gotten to this discussion so far, or yet? For how many of you was it clear? Okay, so I saw one hand and a head. Okay, so for those of you who've run into it in discussion, first, let's just go over what can we conclude from the lithium bromide part of the experiment or outcome. Is normal.
Yeah.
Equal amount of DNA everywhere.
Mhm.
Cool. So now let's go on to the next half. When you approach an experiment, first look for the negative and positive controls. And so what are our controls in this experiment? We talked about in class. What's the control supposed to be? UV minus and our wild type, right? So we have some signal here. That's supposed to be our negative control. And then what's the positive control supposed to be? So exposure to UV. If this is supposed to be positive control, what do we expect to happen when you add UV exposure? There should be a signal. What kind of signal? More or less?
More.
More signal. Why?
You can get a response to
Because on NER pathway. So in response to UV, you should get more damage, more NER pathway repair, more incorporation of the radioactive thymidine. Thymidine. Ha 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 ha. Thymidine, just adding letters to things. It's not for anybody, but feels like it. But what we see if you compare to your negative control, that the signal isn't more, it's less. Why is that a problem? That's just it. It doesn't make sense. This means that your positive control is not working, or it's not accurate, or somebody switched up tubes. So if you have a positive control that is not working or doesn't look appropriate, can you interpret anything else on your gel or in your experiment? No, you can't. So if this is off, you can't interpret the experimental. Okay, so is that clear?
Yes.
So the reason is that you should make the mistake a lot more on a positive UV.
Yes,
Because you see some when there's none, so there's more should be there.
there should be more.
Yeah, I was kind of confused, I'm really just like, it don't make sense,
Yeah,
so therefore it doesn't make sense.
so there, doesn't make sense, doesn't make sense. Yeah.
That's gonna be like, backed by.
Yeah, but you bring that to the right spot, it doesn't make sense. Make it make sense, please. Yeah, so because it doesn't make sense. This doesn't make sense. Okay, good, yes.
Yeah, when there's less like signal on positive UV, could it like indicate like some thing wrong with like the?
This is how I was doing.
But it's wild type, what do you know about wild type?
Oh no, I was just saying like
In general, yeah, but you have nothing to compare it to. And that's why we need a positive control, so you can make that conclusion. So say, for example, this ethidium bromide thing was really, really bright like this. Now, what do you think about this experiment?
It makes sense.
It doesn't, I mean, the results make sense, but you can't really.
The response seemed wrong.
Okay. So now this was really clear. I want to repeat it because he said the results make sense, but you still can't interpret anything from it. So if we can figure out why does the results make sense now?
Is it not because it's too?
Why does that make sense? That's the negative.
Yeah.
When the specters are in the past, we can expect to see the lights.
So this is hypothetical.
Yeah, or this could actually happen in real life, or this could happen in your job. So the ethidium bromide, what does this stain? All the DNA, any DNA that's there. And it's, what does it mean if this band is super big, it's really tiny, and these are really small? What does that mean? There's more. Ethidium bromide has nothing to do with the actual experimental outcome. It's just how much you stuck into the experiment and put on the gel. So in this example, I put way more DNA here. And because there's so much more DNA loaded, you end up having a matching bigger signal, even though it's minus UV. And in this one, I'm like, oh crap, maybe I couldn't quantify my stuff properly, and I put way less. So now I have a small signal. So yeah, so you still can't interpret XP, but now you know why this happened. Yes.
It's like a false positive and false negative.
Yeah, so it's kind of like a false positive, a false negative. And this loading control, which tells us how much DNA is there, tells us why we're getting false positives and false negative. So if I was in a lab, I'd be like, oh crap, I need to do this experiment again and reload so that this is equal. So what we want to do is know that the total amount of DNA is the same for the whole experiment. And then we're able to judge how much new, I guess, thymines were incorporated, because we started off with the same amount of DNA. Yeah, so know what both of these are for.
I was in the garden.
Ask it.
So, for example, when we were trying, that's pretty. And they wanted to say, okay, this is your, like, in gamma rays, will they see, like, for example, they didn't know, this is like, we have just completely, we don't see any, even though there was.
Exactly. So say you did, let's add a third thing here. And let's actually fix this. So that our controls make sense. Okay, so now our control makes sense. And say we want to do plus UV plus sunblock. And say we're using some type of like liquid formulation that we could put in the reaction that will block the UV rays. What kind of outcome do you think we expect to see? So in wild type, we expect a thin line, right? What do you expect here? Still thin line. Yep. Yeah. And so we block what's causing the damage. We're not gonna get damage. We're not going to get more incorporation of time. It's a really good hypothetical. I like it. I turn to an exam question next year. Next year, it'll be an exam question next year. Okay, so now we're here still, yes.
So I remember, it was about today's lecture, we were talking about, mismatch repair,
Yeah.
A and B. I had trouble figuring out, so the reason why we B was because the A next to it has B's.
Okay, show me the photo, mismatch repair.
Um.
Mismatch repair. Oh yeah, this one. TC. Prime. 3 prime. So the whole idea here is that I gave you the scenario. That after mismatch repair, this was the very last nucleotide that got incorporated. Since DNA is always extended from 5' to 3', it meant that extension had to happen this way. This is the very last thing left. This would have had a monophosphate on it, and this has the 3' OH. So this is why this is the gap where there's no connection. Everything else is connected. And if this is the gap, that's where ligase has to act. This window's hot. Burning.
So then that was a C then, but it was a C with that, but B, did that make a difference at all?
No. So as long as I told you that this is the last thing that got incorporated, you know, based off the fact that it has to be 5' to 3', this is where the gap is.
Mhm.
It's after the last thing we added. Okay,
Wait, then as you were saying, like, so then the methylation would be again on like that.
on this side,
Not that, it was made for the methylation by sequence,
the sequence where the methylation would be,
but the actual methylation itself, would also be on the right or just off?
would also be on the left, but it'd be on the opposite strings.
Oh, got
So I think we're here now. Okay, we're on
Yeah,
that.
that's mine.
To the outer rim.
Oh, I had a question from the Democrats last night.
Right,
So, the setup is like we're setting up this scenario and then we have, let's say, the class like the side button. And I understood that we get like the lesson because I have this product, but I just want to know if there's explanation on that.
Okay, so the cool thing about in vitro PCR is that there's a lot of different concentrations of items that are optimized. So salt is one of those that is optimized so that you have good association of the primer with the DNA. Remember, we said that salt is one of those factors that can impact the melting temperature of the primer to the DNA. So the idea is that if we already have an optimized salt concentration, if you increase it or decrease it, we're impacting the melting temperature. And no matter what direction that impact is going to go, it's not going to be good. And what's strange about salt is that different salts are finicky, and there's this idea of salting in and salting out. So salt can make things more stable or less stable. But either way, if you're shifting the melting temperature of a primer, where we specifically design the reagents to work at a specific annealing temperature, we're changing the melting temperature of that primer. And then so it's going to impact the PCR for worse. Hopefully that's clear enough. It's not specifics, but this is what happens with salt if you change something from the optimized salt concentrations. Definitely have to take, we can jump there or we can keep going and come back. We keep going. Oh, question on this.
Yeah, it's like, what other factors would you say like that is like the best piece of product besides the salt.
Maybe-
Messing with the.
Yeah, yeah, if you mess with the pH that you're running your PCR at, same thing.
Or it's like too radically extreme that.
Yeah, it was wild, so it's just like an aside. When I first started learning PCR, there's that transition from people using like these beautifully optimized master mixes. And we had to like figure out what the concentration of magnesium, the concentration of the NTPs, not like, we were still like adding all that one by one to our PCR mixes. And towards the end of my PhD, we got a little bit of money and we started getting master mixes. And then now it's very common, like I don't think there's any laboratory that doesn't use a master mix. So what companies have done is optimize salt concentration, concentrations of DNTPs, concentrations of the polymerase, all to work at a particular annealing temperature. And then you design your primers to match the annealing temperature for the master mix, and then you run PCR. So anything that's optimized, you don't change it.
You're the optimization.
Yeah,
So by changing the salt concentration of pH, you're making the primers non-specific.
no, you're either going to increase the melting temperature or decrease the melting temperature of the primers.
Okay.
And you don't want to do that because the annealing temperature is already set.
We use more salt. Increase it.
It depends on the salt, that's the thing. And so don't stress too much on this. This is a concept. I'm not gonna ask you specific, and I tend to leave salts out of exam questions because they can have different effects depending on what salt. Yes,
Why would you get actually
so the actual mechanism doesn't change. So the DNA is the polymerase is gonna send the DNA the exact same way. Same chemistry, attack the triphosphate, stuff the pyrophosphate, like none of that changes. It's just the temperature at which we're annealing the temperature at which the primer is gonna anneal to the template. Yes, you're here.
So why don't you do that if you can something in there to-
Well, you're not denaturing the actual templates.
Which salt? Not exactly.
Well, we said 2x, it's kind of hard to know. If you put in a crap ton of salt, then maybe you could denature it, but also maybe you could stabilize it. And so that's really, really up in the air, and it's highly unlikely that you're just gonna denature the template with salt. Usually you pull it out of solution before you actually denature it. It's interesting. So I guess technically that could be an answer. Once again, why the salt questions are in homeworks and discussions and not on exams. Here's them here.
Wait, so basically like. Changing condition will alter the mechanism, but I'll just change like what it happens to like, it'll be less specific to what you want it to be. The mechanism would still be the same.
Yes, mechanism's still the same.
So you're changing the annealing temperature, which,
Changing the melting temperature. So you walked in after we talked about melting temperature versus annealing. Annealing temperature is what we're gonna set the PCR reaction at in order to get the primers to anneal to the DNA.
and that temperature is such.
And then we set that
And it doesn't anneal to itself before it anneals.
before the primers, right?
Okay, so then when you add in salt, you're changing.
The melting temperature between the primer and the DNA.
Okay, and then that will affect if it's ready at that temperature to anneal. You know, why this all messes it up.
Yeah, so if you're changing the interaction between the PCR primer and the actual template, then either your melting temperature is gonna be too low or too high. Because we've optimized the salt so that the melting temperature is specific to that DNA template at that annealing temperature.
Okay.
It doesn't affect the annealing temperature, it just affects the.
So it's the melting
Just
temperature.
about.
Yeah, that's kind of an easy way, like, you're just trying to keep it separate in your mind. And just know that a lot of people will use the two words interchangeably, which is what's confusing. Yes, yeah, so, yeah,
But it's just the point of looking at.
the direction, yeah. Yeah, so anytime where there's words that can be used the same in different contexts, I avoid them on exams for this exact reason. So you find yourself getting stuck on a concept like this, you can probably be pretty sure it's not gonna be asked as a question on the exam. It's something I want you to understand and knowing like how these things interact and how they work, especially if you're gonna be working in the laboratory for like designing primers, because it's very, very common. But don't let yourself get stuck between like, is it annealing or is it melting? Just know that changing salts messes up the optimization and trying to run this PCR at a specific temperature to allow these primers to anneal specifically. Ah, see ya. Okay, so we are here, I think, yes.
That's not this one.
Okay. So let's draw this one out again, and you will need to know it. So on the slides, you have a photo or picture of a DNA helicase. You have a long DNA strand. You have probes that are radio labeled. I think it's 796. I think it's 722. This is your helicase. This is your. But which is 3' plus 5'. Under example. It doesn't really matter, but. Wait, so down here?
On the left, it's 3 above, 5 below.
Okay, so I'm going 5', 3', 5', 3'. 5, 3 is 5. You get me every time, always write it out. Okay, so this is what you have as an example with the actual data sheet that we have. What I want to make clear is that when you run the actual experiment, think of the fact that you literally have two separate samples. Say sample 1, and then sample 2. Sample 1 only has the 796 in it, and then the helicase. Sample 2 only has the 722 in it, and then the helicase. And then so the whole idea is that if you have an active helicase and you put it on a template that has a little probe on it, you can't visualize this main piece of DNA that's really long. Only way we can see this piece of DNA is if we see the radio label template. Only way we can see it. So now think of running it out on a gel. You have 2 lanes, one for sample 1, one for sample 2. 796 versus 722. If everything is intact and we have the label on it, where are we going to see the DNA on the gel? At the top, because it's big. So 796, 722, even though the little piece is small, because it's still attached, you see it at the top. If for some reason the helicase is active, it can go from 3' to 5', boop, and knock this off. If we knock it off, then where will we see the actual signal? At the bottom, because it's now small. So this means that we have 3' to 5' helicase. If, for example, we have to go the other way and knock off this one, it's moving from 5 to 3. We knock it off, it's little. This is a 5' to 3' helicase. If you see both, it can go both directions. If you see one, it can go one direction. If you see none, something's wrong with your experiment, or your helicase doesn't work, or is denatured, like something. The helicase does not work. Is that clear? Yes.
So in both of these cases, assuming that it didn't, you would still see an advantage because of your production.
So what's a concept that we talk about in science and experimental biology in general? No reaction is 100% efficient.
Oh,
So when I set it up, not all the probes are going to get knocked off. You're never knocking off all of the probes.
so basically.
There's always some stuff at the top. Unless I said, what if this was somehow reaction was 100% efficient? What would it look like? Which one? Yeah. Yeah, so remember that's the standard. Like, nothing's 100% efficient. See something 100%, something's wrong. Like, like fabrication. Just kidding. Okay. Now we're here.
Okay, so, starting is fair. I think that's no, this about like visualize.
We'll just try and we'll figure it out.
So. The phosphorus remove the base, now you have a BT, right?
Mm-hmm.
And it can into place, cuts it out and brings it up, right?
Yeah.
So the very next thing is slides, so the camera is basically, filling the gap essentially, right?
Yes.
Back. I guess the question that I have is, between the endo and the polymerase effect, how does the uptake DNA get removed? It all gets a little bit in the way of the polymerase.
Polymerase has 3 active sites.
Oh, okay.
Which active site is going to be used to get rid of this blip blip tip?
Uh,
Five prime to 3 prime exonuclease. Think of it like it's like Pac-Man. It's like chomp, 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 and then behind it, it's making more DNA. So the polymerase comes in and cuts this out, cuts out a couple base pairs, while it's also extending the DNA. It's clear?
What if it was on the other screen?
Let me just go another way.
How?
Just the other way. Don't overcomplicate it.
So on the exam, like, how would you, like, how would you, like, ask for, like, you are like, oh, what directions are going to permits and stuff like that, you know?
Yeah, I mean, I probably ask you, it's like, what would happen to DNA repair if it's missing the 5 prime to 3 prime exonuclease activity? Right, so understanding like some of the basic parts and what it does.
So the 3 parts are the 5-degree exonuclease, we have the polymerase itself, that's then the 5 putting on, and then you have the 2-3.
So those 3 activities and what they do. Yes,
But if you haven't been used this stuff, like, want to,
I mean, you wouldn't be able to do this type of repair,
right,
right? So if you couldn't do this type of repair, what happens to the,
oh.
you get more mutations, more damage, right? Like, what's the consequence of not being able to do this type of repair? Yes.
I have a question that
Oh, yeah, okay, we're done with basic decision repair? Bird, sorry. Who said that? Was it? Okay, it was you. That's freaking hilarious. Yeah, I think I'm gonna start saying that somewhere, maybe write a song about it. Okay,
it's going back to the game test that we talked about, last lecture, which was, the final question, which is like, is this compound being tested mutagenic? And it was mutagenic, correct? And how do we know? I just want to make sure I got it right. One of the reasons why we know is because there is an increased growth due to random mutagenesis that causes mutation. So we're getting growth in the form of rings. And then also we're confident because dilutions of a potential mutagen reduce the diameter of the clear zone. That part I was a little bit unsure about, like, what that means. And obviously we're getting rings through the fusion, we're getting the separation. But I was just like that part about that reduction of the diameter.
okay. So, Ames test, we started off with a histidine oxytroph that it's very susceptible to spontaneous mutation or reversion of mutation to that histidine gene. So because it's very susceptible, if you put something that is mutagenic, then it tends to go back and be able to grow all of a sudden on plates or media lacking histidine. So what we see is that if we stick the mutagen in the middle, it forms a gradient of high concentration to low concentration. And in the very, very center, if you have something that's heavily mutagenic, you cause a lot of DNA damage, you kill the cells. Like too much mutation equals death. But eventually you get to an amount of mutagenesis that occurs that results in this reversion mutation. That's where you start seeing this ring, and that's what we call the clear zone. So death, clear zone, then they start growing, okay, reversion mutation. So this is probably mutagenic. So what people do to ensure that this is due to the actual compound is do what we call a dose response. You reduce the doses. We call this high dose, medium dose, and a low dose. You reduce the doses. That way, this zone or clear zone gets smaller and smaller and smaller. So when you see that something is dose dependent, you know it is caused by the actual compound that you put on that disc in the middle. So double verification, you get the phenotype and it's dose dependent. Is that clear? Good.
And that creation of the ring is going back to the idea that if you're getting a reversion mutation, that's where you're getting the ring.
Yes.
Obviously, yeah.
Dose depending and what else?
Just dependent in the fact that you get growth.
Oh, okay.
None of this.
If you just gave us one of these things, so like. Because I thought like the concentration of the mutagen was like based on the gradient.
Yeah, it's based on the grade.
But then do you need, like, if you only get us one of those things, could you determine if it would be glucogenic or not?
Mhm.
Oh, okay, this is still see the dose.
Yep.
Okay.
And so we still see the growth around it. And then the idea, so, okay, so for some reason, let's do. If you have this like ring formation, you can be like pretty sure that, you know, it's mutagenic. But for some reason, you put this. And then you see this splotch over here. Would you be able to confirm that that's mutagenic?
So it's.
Yeah.
It could be like any.
Yeah. Yeah, or maybe you like drip some on the edge or something, or had there's something else that caused these to actually revert the mutation. So what lets you know that it's happening is because you know there has to be a gradient. So other thing, say you had a clear zone, you know, a little bit of growth, but then you had a whole bunch of growth this way. Would you think that this was caused by the actual mutagen? The growth. So we have a clear zone and then there's more out here and it keeps on going. Well, why not?
Okay, so maybe it could be like. You can like, okay, it could be because of the price of them, and I haven't like it, right, from the guy, but the those on maybe is a different, like. They don't know the treatment decision, because the dose is different.
Yeah, so I would have looked at this and be like, oh man, my dose is really, really high. My clear zone is big and keeps growing all the way out to the end of the plate. You know, so you could dilute it to see if you could come back to this.
So.
Could just be like-
Essentially, just the film dim card.
Yeah.
Just film card, yeah.
Like, relative.
Yeah, so just understanding that there's a gradient. High concentration, death, it gets less and some grow, and then it never got like diluted enough or enough space for diffusion that you have another space, you don't see it. Yeah,
Would that be the only thing? Really go up here to go from low to high.
well, technically, it's always hiding,
Sorry, hiding.
it's always hiding,
Okay.
and it's all dependent on the concentration of the mutagen you can put there. And how strong of a mutagen it is. And
The
there's
diameter.
yeah, and so this is the whole reason why people do like the dose responses. Like if I decrease the dose, I should see the killing zone get smaller. And then eventually it should get diffused enough that it's not causing the cell to get the reversion mutation. But just knowing how to interpret it. Any other question on Ames test? Okay. On Ames. Yes. Oh, it's on Ames, is it on Ames?
Yeah.
Okay, let's put it back.
Let me see, Mary, and somewhat lot just a lawn.
Let's do that.
I'm trying to think of like, what could cause that? Not just. I.
So you were probably like last Wednesday or something.
Yeah.
Yeah, okay, so you had discussion, we had this problem on it, it was before we had lecture.
Yeah, they kind of covered it before.
Yeah, yeah,
Yeah.
so Wednesday people kind of got a little bit screwed, so let's.
Yeah.
Okay, so for those of you who had discussion, understood this, in what scenarios would we get a lawn of growth when we're doing an Ames test?
Well, it's not.
Oh, sorry, not an oxytocin, they grow by itself,
Does it?
So you have to start off with the oxytocin,
or you add.
yeah, or we gave it histidine in there, and then they could all be able to grow. So the, go ahead.
Sorry, would that be the only like? Two options, just the.
That's the only two I can think of.
Okay,
They
okay.
have like crazy, like
again.
That's what I was like, I was trying to do like, think like.
No, every mutagen we know because of how DNA damage works, like if you have it too high, you will kill the cells. There's no way to make it like not do less DNA damage even at high concentrations. It's always concentration dependent. Yes,
Especially there was, an option that was wrong, it said that, mutation Y, like, wasn't the reason for this. Yes, and it wasn't a mutation, and I thought that was correct, because like, I thought it was like complete, but it means that like, oh, no, it's wrong, because I. It's like a DNA defective.
yeah, there's no way to confirm, right? So no positive control, no negative control, no like zone that tells you what's going on. You can't interpret what the mutagen is doing. You can't interpret it because the setup was wrong. Controls aren't in place, exactly. Yes,
It was, for example, you are using what is output flow, and then you add your carcinogen. If you didn't get any role, it would be that is.
it's not mutagenic, exactly. So a negative plate is essentially this, not carcinogenic, it's not a mutagen. Yes,
I have a question on the back, this says because we are just on the lower. It was the one in the mutations,
yeah. Okay, so
Okay, so. What, things of mutation, like, lead to, like, cell termination, like.
I mean, technically, so what's the rest of the answer? So this is the practice exam?
Yeah, it's, the going, like, the human cell line, accidentally left it under, oh, you like. Well, we can't. Wait, you recognize your mistake, they're removed from the, unfortunately, all their cells died, except for the solutions that have caused this outcome. I said primarily, communication mutation, because isn't that a stop codon?
Okay, so the idea when you're reading the question and answer steps, when you read the question, what is the actual thing that's being asked of you? Like, what is being asked for you to look for in those answers?
Oh, what mutation is caused from the.
No. The question is actually, what type of mutation is gonna lead to cell death?
Okay.
That's what you're looking for. So is there an area? So I'm looking at broad principles, and partly because not people haven't taken the practice exam yet. So I don't want to just give the answer. But if you think about it, say if I'm looking in an area where there's genes, mutations where there's important genes, that will cause cell death, right? If it's a gene they need for life. So the gene they don't need to live, and mutation there is it's not gonna matter. If you have an area of DNA where there's not even any coding genes, then it's less likely to actually cause an impact and cause the cell to die. So think about the whole entire answer that you see, and then ask yourself, does this particular mutation in this particular area or thing or protein, will that actually lead to cell death?
Okay. And then like, this was just more of like, having of like concept, so like, can I do mutation, like, how do you like, look at the difference on that?
I mean, any kind of mutation that moves cell death. But it just depends on what the mutation's in.
Okay.
Yes.
I have a question going back to the homework that was due, or you were asked that you were saying about PCR reaction. You see the temperature lower than you're supposed to, what effect will this have?
Mhm.
And the answer is that you will get the PCR product from multiple products, not only the products, kind of like what we've been talking about, like temperature on getting. Is it kind of also that notion that like it's not as efficient, basically it's not as efficient now,
It's not that it's not as efficient, it's not as specific.
or not as specific.
You're losing specificity.
So it's essentially set up so that the DNA will bind the primary at this specific
Temperature,
that,
yeah.
like, ensures that the DNA doesn't make it onto itself.
Mhm.
It's gonna know itself.
Yeah.
Okay.
Any other questions? Yes.
So yeah, that's also, it was one where, which one thought I explained why DNA ligase is necessary, and you're like, this is just like clarify. So the answers were like, the mRNA polymerase is capable of forming a phosphate acid bond with a nucleoside monophosphate, which went over. RNA primers must be degraded or phase with DNA nucleotides. This was a bit confusing, like, why is ligase necessary? For the fact that RNA primers can be degraded or fixed with DNA, like why does that prove that we need DNA ligase?
What's the back end of the question?
What?
Is this like a multiple, like, so it's like all that apply?
Use them all.
Yeah, yeah.
So explain why HT ligase is necessary, HT is necessary to form all phosphodiester bonds. But, DNA synthesis cannot occur at the end of a linear chromosome. DNA polymerase is incapable of forming a phosphodiester bond with a nucleoside monophosphate, and the last one, RNA trimers will be degraded and replaced with DNA nucleotide.
Okay, and it's just like all that apply, right?
Yeah.
Okay, so there's multiple reasons why ligase is needed. So not one answer may have like all of the information. And so once again, like when I push you through homeworks, is to get you to think about all the concepts necessary. And then when we get to exam questions, my goal is that you can only think of the answer in one specific way. So dig a little bit deeper, understanding the concepts on the homework, and then should be more straightforward on the exam. And then if it's not, let me know and I fix it, give you points back. Yeah, I get 3 points.
Okay, so is the reason why RNA trimers being degraded explains the reason for DNA ligase is because of that awaken or?
No, so the whole process of like the fact you have to go back and get rid of the primers means that you have DNA polymerase filling the gap, but then now there's a gap that the polymerase can't ligate. So it just all adds into the overall big picture.
Got it, okay,
Yeah, so-
that was like very specific, but I guess.
No, yeah, so it's done with that trip you up.
Okay, got it.
Wait, sorry, where's the gap to me exactly?
Oh, so you know when you have like the primer and you synthesize the DNA, and then you get to the other piece of the DNA that's here.
So the other primer?
Yeah, so now you have to like degrade the primer. The polymerase one, so Paul one comes in, degrades the primer and adds it, and then also now we have this gap here. So now ligase has to fill it.
Oh, because it doesn't have the
Yeah.
phosphate.
Yeah.
Oh.
So it's just having you like mentally walk through the steps.
I was wondering why DNA synthesis cannot occur at the end of the chromosome, why that is wrong. Is that just because like you have like telomerase and all that?
Well, DNA synthesis can occur as long as there's like a primer there. It depends on
so
what
if it's, like,
side.
on the leading
Yeah,
strand, then it's
Yeah,
fine.
that's fine.
Oh, okay.
And the other strand, no.
So
Yeah.
I'm understanding.
Yeah.
Okay.
I have a question for those from that, so this is, I'll be the whole thing, so imagine a cell had a version of helicase that was permanently stuck at the origin of replication. We consider a dominant negative mutant and why? And the answer was yes, since it will prevent normal helicases from functioning. But did we go over that ever with dominant mutation?
No, we didn't. So this is a concept that hopefully you learned in genetics. And I
Not as much helicase, well, I guess.
yeah, but like dominant, negative,
Yeah, yeah.
and so I try not to put the types of question on the actual exam. But it's information you should have carried over from genetics, but if it's dominant negative, it's basically doing a function that stops the minor allele from being able to function. So essentially, you have your origin of replication, changes the positive, you have a helicase, it's stuck there. So even though you have like 2 alleles, and say one of them is like a regular helicase, other one is the effed up helicase, helicase is gonna bind here, the other one can't get on. So it's dominant negative. Even though there's only one allele of it, you still see the phenotype from the expression of this helicase.
So dominant negative is.
I mean, it's yeah, it's dominant negative because it's not homozygous. Does that make sense?
Okay.
So usually what you have, if you have something that's, now we're doing genetics now. Genetics. Let's go with wild type helicase is normally what you have. And then say we have this weird thing where you have the wild type and then the mutant. Usually what happens is that the wild type will just take over and cells will look normal, even though you only have one copy.
Correct.
But since this mutant is dominant negative.
Yeah.
It's gonna overtake what the wild type's supposed to be because of the way it functions.
Gotcha, dominant negative, mutant overtakes wild type.
Yeah, this is not part of our class, it's genetics, so just let you know.
But when overtakes the wild type, that's why we don't, therefore prevention of normal function.
Yeah, so in your mind, if you hear dominant negative, it means the mutant, that's the phenotype you're gonna see in the cell. Yeah.
So in this example, are there other cases that like, there are different types of helicases that aren't mutated and it's dominant negative because you have one that's preventing?
The others should lose their function.
Oh, okay. Even though it only affects like one.
What one, yeah,
Oh, okay.
exactly. Well, the reason why I know this stuff, like my friend's a geneticist that I did my postdoc with, and she started talking about like dominant negative things and like truncated proteins. I'm like, what is going on? So I learned a lot from her research project and her being in debate like next to me. Oh, yeah, it's just some really cool stuff, really cool approaches in her lab. Yes.
One more from the quiz for this quiz lecture was, results always not true regarding patients with pseudo or pigmentosa. And the one that's false, because they have a faulty base excision repair pathway. Oh wait, it should be a new, it's not like, it should be
Yep. NER,
NER, okay.
yeah, the burner,
got it. NER.
yes, so keep those three straight, mismatch. Base excision, nucleotide excision, know which ones they are for and who they relate to. Do them straight, if you have to, write out a little cheat sheet for yourself. Okay, any other questions? Yes.
I was a little confused on like, I guess how the trombone family, like, in particular works.
You and me both. Okay. Yes, what I can say that combo model itself is not gonna be on the exam, but there's a video that I have that helps to demonstrate it. Apparently, it's not active on Canvas. Someone came to me, I can't find a trombone model thing. I was like, oh my God, my bad. So I'm gonna activate it so you can see it. But essentially, the whole idea of the model is that you have a reposome that has 3 polymerases actually associated with it. One of the polymerases is going to be on that leading strand just doing its thing. And then the other one, they're all attached in one big like protein complex, is pulling like loops of the actual DNA so that it can replicate the lagging strand, but keep up with the replication for it.
So they're all moving at the same.
So they're all moving at the same time. So it's loop, replicate, loop, replicate, loop, replicate. And so the video kind of helps you visualize it a little bit. And that's what the trombone model is. So they think of the loops as like that arm of the trombone as you're playing. And that's the way they're able to keep up the replication on the lagging strand with what's happening on the leading strand.
And then I guess I'm thinking these somehow telomeres work and like telomerase reverse transcriptase, like. So are telomeres like, are they just allowing themselves to like not be replicated? Like that's the point of them. It's like WDNA that like it's okay on some use.
Basically, it's like, think of it as buffering protective ends. They're just repeating sequences that don't mean anything to the cell. So every time they divide and they lose it, it's okay to lose it.
And then what builds it back?
So what builds it back is telomerase, and telomerase is in cells that basically need longevity. So think about your germ cells, when they make your eggs that keep populations alive and take genetic information from generation to generation. They have telomerase to make sure that they protect all genetic information by elongating those buffer zones. Like they still get chopped every generation, but then in those cells, they can rebuild it.
So does it just rebuild it from double stranded DNA? Like,
Yeah,
or-
yeah. So say like telomerase, you have this 3' overhang. But. And then because the whole like end replication problem where you get rid of the primer, like telomerase just can't come in here and make anything. So then what telomerase does, it just makes its overhang hella long. Like extend. Then you can drop the primer down somewhere and then fill it in with DNA with polymerase.
So you're dropping the primer and all the DNA that you want, and then to the right of it is just more
Repeats.
repeats. Okay.
So 5', we'll say this is 3' overhang. This is gonna be our DNA here.
And that's the DNA repair that.
So this is the DNA we care about.
Uh-huh.
Telomerase is gonna come in and then add this, make it really long, and these are all repeats.
Oh, so that's what it wants. Oh,
Yes.
so it physically adds it.
Physically adds it, makes it just really, really long for repeats.
Then you drop them.
And then we can drop a primer over here. And then polymerase. Can fill this in.
Oh, and then you'll have a name, so then. I
So now we have a buffer.
Oh.
Okay, cool. Any other questions? Yes.
Regarding the product rating for today. It's like, if there were a couple questions at the end that seemed to not be part of the assignment. I'm not sure.
Oh, yeah, I think part of them were for next week, so, my bad. If there was an issue with like, cause I mean, you were able to submit it 3 times, so hopefully it shouldn't affect your grade or anything, if you saw the answers. But for the reading assignment, I think the last couple were off of some of the reading for next lecture, and I think I was pulling some of the material back so that we could focus on majority of topics I wanted to get through, so there's less time between new material and exam. That way, there's like only one major topic to hit on Tuesday.
There's an experiment, and it's like, you got 2 names in there. It's not
Yeah.
lots of- like, talking about it. but. But like, part someone else, I don't know. So like, I searched those names in the book, and they weren't in the book at all, because they're outside of the assigned meeting, so to me that makes me think that it wasn't any of the assigned meetings.
Okay, so what was the question? So I went through the reading quiz before I posted it a week and a half ago. So just remind me of the question.
Yeah, what was in part, and then by regulations, details, that don't apply. What involved? Maybe something like.
Okay, so what's the information for, just read me the question.
What was in part, identify regulations, use result that so don't apply, and so individual, the combination to this common that cannot go on the and the consumer, this common that you don't without that.
What mutant does the word before the word mutant?
Like, I see.
Okay, so you missed the definition for rec A in there. Okay, I'm gonna double check that question and make sure it's just not like outside of scope. Because usually I try to give enough information so you can answer the question, even if it's something not in the reading. But I can miss things. So I also have like a bank and I'm adding questions every year and I pull from the bank. But sometimes I tell it take some like random ones from the bank. And it's probably one that I just missed. So he says question 12, reading quiz for this week, for today's lecture.
Yeah,
Okay,
so like, I searched the book, for another than part. And it said, well, we found these names and they're on page 479,
yeah,
but we don't have more stuff.
79, so it's probably one I just like cut out, so don't read this time. So I'll go check that one.
But so we wouldn't expect those to be covered at all, because
Nope,
if it like, even like in next week's.
nope, that's not much, definitely not. I prepped for Tuesday this week, so I'm like, no, it's not there. Yes,
For the practice test, there's like, rewritten questions, is that supposed to be there, or are they supposed to be different sections?
I'm pretty sure different questions on the same topic. I'll double check it and make sure it's not like a glitch where questions got duplicated.
I think it's like the last.
Okay, so I'll go check them. Yes. Here, okay, here.
This is, I mean, we like showed these in class, I was just wondering like what these two were doing. Is it?
Oh, they're helping to identify where the thymine dimers are, but you don't need to know.
Oh, okay, you just like mentioned knowing what they did with
Like, just know that some protein binds, about 2 nicks on the side, gets rid of the DNA.
okay.
Yes, oh, we had to go here.
Yeah, if you guys rapid fire, use quick questions that have the reading, that I guess you're going to.
I mean, okay, go ahead and try. Let's see, I don't know if my brain's that rapid.
And it was the one with deamination, oxidative damage, oxidation, all that stuff. If it wasn't mentioned in lecture, do we know when in the cell cycle each of those occur or not really?
No, not really. What's important is going to be to know when in the cell cycle you use certain types of repair mechanisms, specifically if you're comparing like homologous combination versus non-homologous end joint.
But, and then the same for all the, you talked about some repair mechanisms are when the cell is being lectured, but the ones in the reading, we don't really worry about too much unless if it wasn't mentioned in lecture. We talk about cell cycle and stuff with one of the.
Yeah.
Just focus on that.
Yes. Okay, we go here while you're looking.
Yeah.
Could you like briefly explain which lobbies assays were, sounds like some like, what they measured?
Okay, so we're, cool. We're gonna walk through it one more time, but you need to know it. You
Yeah.
need to know it. And so remember we said that telomerase is gonna extend that 3'.
Mhm.
Telomerase has its own RNA primer. That's one important part to know. This RNA primer is a certain length, so it adds on repeats of length. So when you run it on the gel, so you start off with a exogenous DNA. As you add on repeats, you get DNA pieces of different sizes, which is why you see this whole entire like ladder of DNA when you run it on a gel.
Sorry, as you add on repeats, you get different sizes.
Yeah, so say one piece of DNA, I added on 1 repeat. Another piece of DNA added on 2 repeats.
So this from like different cells, different replication.
So from one cell. So it's depending on telomerase, like which piece of DNA it jumped onto and extended, it's basically random, it's stochastic. We weren't, I learned that on one cell. So essentially you start off with short DNA that we put into the assay. And we assume that there's telomerase present, so telomerase present. With the right conditions, we let this telomerase go ahead and extend DNA pieces. Some of the pieces, it'll extend and make it super long. Some pieces a little bit shorter, So you get these different lengths.
But doesn't it have to extend at a certain length in order to like save the genetic?
In an in vitro assay, it's gonna extend as much as you allow the assay to run for.
Oh, okay. But in the body, it knows like.
In the body, it figures out when to stop. I don't know how it figures out, but it tries to get it to a certain point where there's buffer. But even then, there's a lot of variability in how long how long the telomeres are.
Got
It's
it.
a range.
Uh-huh.
So on the gel, you're seeing these different sizes, the small ones being at the bottom and the bigger ones being at the top.
Oh, okay, so like in this experiment, when it says RNase was added to show that it was template independent. So RNase would be creating RNA.
Yeah, where's the RNA in this experiment?
In the telomeres.
Mhm.
So it degrades that, so then.
Now it can't do anything.
But how does that prove? Oh, no, it proves template.
Dependence.
Oh, okay, so
Right.
it needs.
It needs the RNA. It needs the RNA in this telomerase for it to do the function.
And then how does that, I guess, relate to the whole like cancer versus non-cancer cells? So like cancer cells are just, they would have more telomerase species?
Yeah. Well, who expresses telomerase and who doesn't?
Um,
So this
human cells.
Yeah, so this is the thing to know. Cancer cells express telomerase, stem cells express telomerase, germ cells express telomerase. Things that are terminally differentiated don't normally express telomerase. So like
Got
skin cells, immune cells, cells that are like part of our adult bodies.
Mhm.
And then so telomerase expression is normally reserved for things that have to propagate genetic information across generations.
Mhm.
But then you have things like cancer, where it's abnormal expression of telomerase. Because the cancer wants to rapidly divide, they hijack the whole system to protect their DNA so they don't die.
So it extends it even more, that's why you have.
Yeah, just keep, exactly.
Oh, got it. Thank you.
Yes, back to your question.
Yeah, the one with the main idea between how chemotherapy and what's basically how they target rapidly dividing genes. I think
Mhm.
That's the main idea you would see wants to take from that.
Yeah.
And then, which was, how do these, mismatch repair identify the gene to replace? Was it, itself, the strand that we talked about, this is the one methylated strand.
To mismatch repair, you have mismatched bases. Because the bases aren't mismatched, you get a structural change in the actual DNA. The
And then where's
base
it
pair-
methylated from?
So in a prokaryote, it's basically going to be methylated on whatever was the older strand or parent strand. So methyl groups on the parent strand, and there's a lag because the damn methylase hasn't come through yet
Oh,
on the daughter
okay.
strand.
Yeah. Okay. And then the next one was like, how does MMR differentiate between a newly synthesized strand with error versus a parental template and prokaryote and eukaryote? Oh, wait, we just went over this.
Yeah, so in in eukaryotes, it's basically all the nicks.
Okay.
So any places where you have to like fill in with ligase, it helps it figure out like, okay, this is probably the daughter strand. The mechanisms still aren't entirely clear to scientists. We don't really know. We kind of think we know. We're guesstimating based off of information that we have. But in prokaryotes, it's very clear that it identifies the methylation.
Got it. And then when it comes to this, what determines the specificity of uracil DNA glycolase, like why it's important and what chemical reactions they normally counteract. I guess it's mostly for that basic tissue repair pathway. In a
Mhm.
sense, and they check, and like the chemical reaction is just like the whole cleavage process of removing and replacing what's going on. It basically just follows the BER pathway.
Yes, so it's BR pathway, just know that each glycosylase is specific to the base that is going to cleave.
Yeah, so it counteracts the single base.
Yes.
When it comes to the recombination process relies on many of the DNA replication principles we discussed, described how, is this about this idea, how like, when a double strand break occurs, it requires an undamaged source of duplicate genetic information that is homologous and double strand DNA. The broken end are processed to generate 3' single stranded extensions, and both ends are used for strand invasion of the homologous double strand DNA form like loops. The invading 3' ends are extended DNA polymerase, just like in DNA replication, everything else follows suit, like DNA replication.
Yes, and so what I'll say for the mechanisms for homologous recombination, non-homologous end joining, don't let yourself get wrapped up trying to memorize all the steps. The most important things are going to be the differences between the two and when the cell chooses to use each one. You can always look up the steps in your textbook and in your notes.
Got it.
And so that's not the concept that I'm trying to drill into your brains.
And then my last question is the one, is actually on what, you brought up with the Margellus and Clark. So I did like some separate research on that. I don't know if I agree what I read on that or just.
Just let it go.
Okay,
Let it go.
all right, all good, thank you.
Okay, cool. So remember, office hours on Tuesday after class, if you're available, exam review on Wednesday, let's questions next exam. We have to let you guys go. Dr. Inlay, treat him good, y'all. Like, I'm serious. Like, he cares. Good person, good people. Okay, so I'm gonna shut this down and we'll see y'all next week.